Hello everyone, welcome to my channel. And this was my first video. And first of all, I introduce myself. This is Sharavardhini, pursuing first MSc Microbiology, Sacred Heart College Autonomous, Tirupati district. And here we're gonna see the difference between the capsule and slime layer. These capsule and slime layer was present in the outer covering of a bacteria, and it's composed of some components and elements, and we're gonna see it. First of all, we shall see about the glycocalyx and what it is about. Glycocalyx. It is a gelatinous sticky substance that surrounds the outside of the bacterial cell wall. It's also called as a sweet cup or sugar cup because it composed of a polysaccharide or polypeptides in the layer. And now here we see what is capsule and what is slime layer. First we see the capsule. When the glycocalyx is firmly, which means strongly attached to cell wall in outside of the bacteria, is called capsule. It's usually gummy, slimy and sticky nature. Capsule is usually made up of polysaccharide, but in some cases of bacteria like Bacillus anthracis use poly-D-glutamic acid to make a capsule layer. And now see the slime layer. The same glycocalyx compound which contain glycoprotein molecules are loosely or weakly associated or attached within the cell wall is called slime layer. This layer composed of glycoproteins, glycolipids and exopolysaccharide. And now here we see the differences between capsule and slime layer. First, its attachment. Capsule is usually firmly attached, which means strongly attached to cell wall, but slime layer is loosely or weakly associated with cell wall. And second off, it's made up of polysaccharide. Capsule usually made up of polysaccharide, and slime layer it's made up of glycoprotein, glycolipids and exopolysaccharide. And the next is its character. Capsule is usually thick and high rigid than the slime layer, but slime layer is thinner and less rigidity than the capsule. Next, its nature type. Capsule is hydrophilic in nature, which means water loving and non water soluble. But slime layer is hydrophobic in nature, which means fear of water and water soluble. The next is its antigenicity. Capsules are antigenic in nature, which means it's able to cause disease or infection to the host. But slime layer are not likely antigenic. The next is its layer. Capsules are well organized layers, but slime layers they don't have a well organized layers. They are usually unorganized layers. And the next is its character. Capsule is usually thick and high rigid than the slime layer, but slime layer is thinner and less rigidity than the capsule. Next, its nature type. Capsule is hydrophilic in nature, which means water loving and non water soluble. But slime layer is hydrophobic in nature, which means fear of water and water soluble. The next is its antigenicity. Capsules are antigenic in nature, which means it's able to cause disease or infection to the host. But slime layer are not likely antigenic. The next is its layer. Capsules are well organized layers, but slime layers they don't have a well organized layers. They are usually unorganized layers. And now its presence. Capsule present in some kind of bacteria, but slime layer mostly present in wide variety of bacteria. Next is antigenic substance. Capsule contains K and B antigen, which is responsible for its antigenicity. And slime layer doesn't contain any antigenic substances. And now its staining property. Capsule can be stained and visualized by using nigrosin or Indianic, which is a acidic stain. But a slime layer visualized by using methylene blue, which is basically a basic stain. Next is a microscopic visualization. Capsule appears clear halos surrounding bacterial cells under the microscope. But slime layer appears a fuzzy halo which extends beyond the surface. And the next is where it is found. Capsule are most commonly found in gram-negative bacteria, but slime layer found in both gram-positive and gram-negative bacteria. And some examples of capsule are Klebsiella pneumoniae, Streptococcus pneumoniae, and Pseudomonas aeruginosa. But slime layer, it consists of Staphylococcus species, Bacillus anthracis, because it contains only D-glutamic acid. And now, here we can see the view of a slime layer and capsule under electron microscope. Here slime layer shows a fuzzy halos around the cell, and here capsule shows a clear halo around the cell. Now here we can see the functions of a capsule. The major function of a capsule is adhesive power, which means attachment. It acts as a major virulence factor for bacterial disease causing. And it prevents bacteria from phagocytosis, which means cell engulfment or cell eating. It also offers protection to bacteria from various issues like desiccation, extreme dryness, hydrophobic toxic materials like detergents, and the bacterial viruses called bacteriophages. And here the functions of a slime layer. Likewise, a capsule, slime layer also plays a major role in adherence, which means attachment of bacteria of a solid surface. It provides also protection too from various issues like desiccation, extreme dryness, and loss of nutrients and antibiotics, and from the cell wall degrading enzymes. And here I gave some glossaries to make you to understand better. They are glycoprotein, glycan, which means sugar, and plus proteins. They both together to form a glycoprotein. And the next is hydrophilic water loving capacity. Hydrophobic bacteria, which has a water heating capacity, which cannot live in the water. And phagocytosis, which means a cell engulfment or cell eating. And the clear halos, a clear circle of a light shows around the bacteria, which usually is found in a capsule, and fuzzy halos, blood circle which less light around the bacterium, which usually found in the slime layer. And that's it for today. I hope you were all understand about what is capsule and slime layer and the various differences and the various functions of a capsule and slime layer. Thank you one and all.